The United Nations Sustainable Development Goals are a comprehensive and collaborative result of the 2030 Agenda for Sustainable Development, established in New York, September 2015. The UNSDGs represent a global partnership of UN countries that recognizes that achieving improved health, education, and economic growth is synonymous with reducing poverty, inequality, and the effects of climate change, all whilst preserving our natural world. Goal 15, Life on Land, seeks to safeguard, restore, and encourage the responsible usage of land-based ecosystems, manage forests sustainably, fight desertification, stop and reverse land degradation, and prevent the loss of biodiversity. In particular, our chosen target, Target 15.8, focuses on reducing the impact of invasive alien species on land and water ecosystems by implementing actions to prevent their introduction and control or eradicate the presence of priority species entirely.
Molecular biology links to SDG target 15.8's aim of introducing measures to prevent the introduction and significantly reduce the impact of invasive alien species by providing essential tools for detecting, monitoring, and managing invasive species. Through developed understandings in genetic and DNA replication, more specifically the Biology 103 learning outcomes, techniques are developed and subsequently used to help identify invasive species and track their spread. Some of these techniques include genetic analysis, in which the DNA of organisms is extracted, sequenced, and examined to study their genetic makeup, DNA barcoding, in which short standardized sections of DNA are used to quickly and accurately identify a species, and molecular diagnostics, techniques such as CRISPR-based tests to detect specific DNA/RNA sequences associated with species or pathogens. These techniques, or relating to the topics explored in Biology 103, can be subsequently used to help identify, track, and limit certain invasive species and their spread. Additionally, genetic engineering can support the development of targeted control strategies such as knockout genes and gene splicing. These approaches improve understanding of ecological and genetic interactions and enable effective prevention, control, and eradication of priority species.
CRISPR-Cas9 is being researched and developed as a non-lethal gene editing tool to control invasive alien cane toad populations by generating less toxic toads, as well as creating Peter Pan toads that cannot grow to adulthood. So how does it work? CRISPR-Cas9 is a gene editing tool that works like a molecular scissors. It consists of two parts. Cas9 enzyme, a protein that can cut out DNA at a specific location, guide RNA, a short RNA molecule that acts as a guide, directing the Cas9 enzyme to the precise target DNA sequence. This method allows for modification, deletion, or insertion of genetic material. Scientists use this cut and paste technology to study gene function, create disease models, and develop new therapies for genetic disorders. CRISPR-Cas9 gene knockouts is currently being carried out by research teams such as Professor Rick Shine's Team Buffo, where they are focused on disabling the gene that enables cane toads from transforming from a tadpole to a toad, and creating toads that don't produce much poison and aren't very large, to create teacher toads, to educate vulnerable predators to not eat toads. The CRISPR-modified toad is much safer than using normal toads for this purpose because a mutant toad is toxic enough to train predators but not toxic enough to kill them, even if it survives to grow old.
The target indicator is measured by the proportion of countries adopting relevant national legislations and adequately resourcing the prevention or control of invasive alien species. Many nations now have put such legislations in place, often integrated into broader laws covering sectors like animal health, plant health, fisheries, and aquaculture. According to a recent report from the Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services, around 80% of countries include invasive species management in their national biodiversity targets. However, only 17% have dedicated laws or regulations, and nearly half provide minimal or no funding for their control. To meet the goals of the SDGs, it is crucial to reduce the rate of the new invasions by 50% by 2030, with priority given to sensitive regions such as islands. Strengthening legislation, funding, and coordinated action will be key to protecting ecosystems, biodiversity, and communities for the future.
A report conducted by Thasinger and collaborators found that genomics serve as highly effective tools in identification and modification of genomic regions to increase fitness, persistence, and adaptive potential of endangered species. Thus, scientifically informed management decisions can be made regarding elimination of invasive species. We consider how this notion could be applied to invasive species to have the opposite effect, that is, using identification and modification of genomic regions to reduce fitness, persistence, and adaptive potential. Ultimately, molecular biology provides the basis of our understanding of life on land from a small-scale genetic sense and also the bigger picture of species biodiversity as a whole.